So this is how you extract your sorghum out from the roots. So first what we've done is any of the plants that are too short or in comparison to the other ones, we've plucked them and set them down. So that way when we're going through to get the plants, that we're making sure that they're all 12-day after planting. So first this is one of the ones that works. We have our 3 leaves. So what you want to do is you want to take your finger, and what I do is I dig to the bottom to make sure that we're underneath all the roots, and then I pull it up. I'll break off some of the other big chunks of dirt. Again, trying to not cause a ton of stress to the roots. Kind of shake it off, get off the dirt. Again, we want as little dirt as possible because that will make our dissections go a lot smoother. I'm going to take it over here to our water, shake it off in here, and put it in here. And then now we'll continue to another one. So again, I'm going to take my finger, I'm going to dig it to the base of the pot. Again, not to disturb the roots. Gently shake off the dirt. And after I get a good amount of the dirt off, I can put it in the water. And after we do a handful of these or a lot of these, then what we'll do is we'll empty out the water and throw out the dirt and put it in a new container where there is less dirt in the water. Because as you see right now, is even though I'm trying to get off as much dirt as possible, when I put it into this container, there will still be dirt that's shaken off into the water. And again, the cleaner it is, the easier the dissections will be and the less likelihood of having contamination.
Okay, so we've got a bottle of ethanol here. You just spray down, I already sprayed it down, so I don't want to spray it again, but you can see the water on there. Not water, ethanol. I just wipe it down. You just want to make sure your surface is clean. You don't have to be crazy about it, but you don't want stuff floating around that'll contaminate it. And we use special wipes, Kimtech wipes, like you use in a lab.
So it's really important with our liquid nitrogen that we're making sure that it's filling our boat completely, and so that way it doesn't evaporate as fast and that our buds will stay at a very low temperature and frozen, and that they won't change their sequence and processing. So you're going to pour it into the side, again making it so the green thing will start to float. Then after that, we're going to use one of these boats, again making sure that it's clean, that there's nothing in there, because again we don't want cross-contamination. We like to bend it, dip it in, and you'll pour it into your container. But when you're pouring it, make sure that you don't pour it so it overflows, because if it overflows, then your buds can fall out. Alrighty, so one of the things to notice is within our test tube, first of all, it's snapped off, and that's okay. But within the test tube, we have holes here. And it's very important that you have those holes, because when the air freezes and it's at very low temperatures, you don't want it to explode or contract and make it so you can't open it later on, or that it won't explode when you put it in the freezer.
Okay, we're going to talk just a little bit about seedling biology and how that works with the dissection. So here we've drawn a diagram of what a seedling looks like and labeled some of the parts. So there's this kind of pseudo leaf that it's called the coleoptile. It's at the base of every little seedling. This is going to be the first thing that along with the roots, you need to remove the coleoptile and the roots because they get in the way when you're doing your dissection. So I always just pull it off by hand, and then I take my razor and I chop it off right there. And you want to cut it off just about right there, otherwise the roots will get in your way. But you want to hang on to just a little bit of the roots, because oftentimes you'll hang on to the roots with your tweezers and be working up here. If you try and hang on to the tissue up here, it's much more gushy and you'll just destroy your little seedling. So you want to leave a little bit of the roots so you can hang on right there. Anything to add, Tori?
Also, if you have dirt on the roots, it's important that you're shaking it off in the water, because you don't want any cross-contamination and it'll make it easier to see.
So keep it clean. Now, let's just talk a little bit about how these grow. So, looking at this diagram, it's kind of hard to describe how they grow because, from what I understand correctly, the topical meristem, the part of the plant that's growing up, if that's not correct, is in the center, and it keeps producing new leaves here. And so the oldest leaves are the ones on the outside. And so the leaf that we're looking to, the bud that we're looking for, is at the base of the oldest leaf, the first leaf. And you'll also notice that one leaf covers the entire stem, and there's a section of overlap. And so in whatever method you're using to unwrap it, generally you kind of want to come in from the side that's overlapped and come in over there, and then come over here and knock it off, and then when you pull off the stem, you'll see your bud right there. Anything to add to that?
Yeah, noting that the shape of the stem is that it's oval shape and you have your two sides, and your meristem will always be at one of the tops of the oval. And so let's say you are turning this around, and you know, if you were to start here and you're looking for right in through there, if you don't see it, then it'll always be on the other side of that same place.
So a common technique that a lot of us in the lab do is once we remove the coleoptile and the roots, we chop it off right here. And unless you start from up here, it's kind of hard to find this spot over here where they overlap. So that's what Tori's talking about when you can use the fact that it's kind of ovular in shape. If you say you end up cutting right here and you're not sure which way to go, you can find either of the ends, and if there's no bud here, then you know it's on that side.
And it tends to be the thicker end that it's going to be on. And again, if you do that by sight and by feel of the needle, and if you first guess of which one you think is the thicker end is wrong, then again, it will be on the other side.
And so this, just connecting these two diagrams here, this bud will always be right here. So it's kind of on the opposite side of where the coleoptile is. And at the base, it's on the same side of the first leaf. So the coleoptile will be on one side, the first leaf will be on another, and the second leaf will be back on the same side as the coleoptile. And for me, it's always easier to start down at the top and cut down either side, so I know where this is, because then it's easier to find that. But what most people do in the lab is just chop it off right there and then come in at some spot and find the bud.
Yeah, one of our lab members works where he'll find the spot right here and take his needle and just pull up, and then pull and unwrap the leaf. However, for me, what I tend to do is with the oval apica side, I'll cut down the leaf from that side, and then I'll do it on the other side as well. And then after I cut those two sides, then I'll pull off the leaf, again cutting then now from way below to make sure that I'm not disrupting where my meristem will be, and being able to pull the leaf all the way out of the way.
Here is a diagram when you've already removed the first leaf. This is the second leaf. So the first leaf has already been removed along with the coleoptile. And actually this bud is in the wrong spot. It would be over here on this side, if I'm not mistaken. But you'll notice that it's kind of arrowhead shaped, and it's just this little thing that sits there at the base. If you were to look from the side, so this is rotated, you'll be able to tell that it is the actual meristem that you're looking for, because it'll be separated from the stem by just a little bit. So what you can do is you just come down with your needle and you plop it off, or sometimes you might have to cut a little bit from the base and you'll have it on the tip of your needle. Something that's really important, there are a lot of other little nubs and other types of tissues that probably develop into something at the base. So make sure you're finding the thing that looks like an arrowhead and that's separated from the root, or not from the root, from the stem by just a little bit. So just a review of the steps. You'll want to remove the coleoptile and the roots and make sure everything's clean. Cut off the roots and then do something to get rid of that first leaf, and then remove the bud. And that's it.
Alrighty, so for the dissections of the sorghum, we're going to take off the first coleoptile. Again, making sure it's good. And again, it works best if you have it as clean as possible so you don't have any contamination. What I do is I then take it and I cut above, probably about a few centimeters, not much. We take it and I'm going to put it underneath here, and when I put it underneath here, in one hand I'll have my tweezers, in the other hand I'm going to have my dissection tool, the needle. And what I like to do is, so the way that the shape of the stem is in such a way that it's an oval, and so the meristem is either going to be on one of the sides of the, not the sides, but the top or bottom of the oval. So I'll cut one of the sides and then start to unwrap it, and it's going to be underneath the thicker leaf. So I picked the side that I think the thicker leaf is on to then move it. And I find that when you're cutting from the bottom off, it'll make it easier to pull the leaf back. So as I've pulled back the thicker leaf, I can see my meristem. And so what I do is taking the needle, first I'm gonna move the leaf out of the way so that doesn't cause any problems when we're dissecting. I'm gonna take my needle and I'm gonna take it to the top of the meristem, right behind it, gently going down as far as I can. Again, not to break or disturb the meristem. Then I'm going to cut right below the meristem as well to pick it up. And then now I'm going to be able to get it on my needle. And when we put it on the needle, you want to try to make it so it's as little in contact with the needle as possible. That will make it easier when you're trying to put it in your test tube. So once I have it on there, if you can see or not, it's at the edge of there's a little triangle at the end. I'm going to hold this green thing down again. I don't want it to submerge all the way because then it might come out. I'm going to take my bud, put it in. It'll bubble as it's freezing and getting all of the excess out. And so as you can see, it just froze. And it's still just at the tip of my needle. So then that way, when I'm putting it in here, I can just snap it off and then we'll be good. And that's how you dissect sorghum.
Okay, so here we've got our plant. You can see it's got 3 leaves, and it's got the coleoptile at the base. So I'm halfway through removing the coleoptile. But I always pull that off first, and then once I've got it off, you pull it off because it gets in the way. If it's like, you'll notice when you're dissecting that if you haven't fully removed the coleoptile, sometimes it gets in the way of you removing the first leaf. Because if you remember, you're basically peeling off the outer layer that's associated with this first leaf down here at the base, because you want the bud that's right at the base of that first leaf, because the buds come on either side of the stem, and the bud that you want, if you find the first leaf, it'll be right at the base of where that first leaf is. So you remove the coleoptile and then take your little razor thing and you get rid of most of the root. This is how you're left with the plant that looks like that. And I do mine a little bit differently. I generally hold it by hand. I gotta adjust my microscope here. Hold it by hand, and I take the so the needle, it's got a point to it, a slanted point. So I take the dull end and I run down the side of the plant just to kind of open this up, and then when it hits the base, I cut at the base. And so I might do that twice here. So I cut at the base. So essentially I've cut maybe 1/4 of this first leaf. And then I'm going to go to this other side and do the same thing. So just run down this leaf, kind of separating it away from the stem using the dull end. And then when I get to the base, I cut it. And so what that does is basically half of the leaf is off, and now it's really easy to just pull it off. There it goes. And I always see right at the base is where the meristem is. And this one's pretty small, but you can see it pokes away from the stem. And so there you go, got it on the needle. I'm going to put it on the end of my needle to make getting it off in the tube easier. Okay, and then liquid nitrogen's over here. So what we do is you put it in there and you let it get to a solid phase, and then you poke it against the edge until it pops off. And it's still on there. If you're not careful about where you place it on the needle, you'll do what I did this time and you'll have a hard time getting it off. And you don't want to put your needle too far in there, otherwise you'll risk picking up other little buds that you've already put in there. I think I finally got it off. So I always run back to the microscope, and I put my needle back under the microscope to make sure that I see no bud there, and I know that it went in the liquid nitrogen. That's it.